There's a famous quote which goes something like, if you cannot measure it, you cannot improve it. Funny enough, this stands absolutely true for aging. How do we measure aging? The number of candles on your last birthday cake? Or something deeper? And can we have better measures of aging that actually help us build better and newer longevity interventions? Hey everyone, welcome back to Decoding Longevity, where we break down the science of aging and longevity. I'm your host, Raghav Sehgal, with my co-host, Macsue Jacques.
Hello, everybody. In our last episode, we talked about what the hallmarks of aging or signs of aging are. But today, we're going to be talking about how we can quantify the signs of aging in our bodies and can they be used to identify longevity interventions? For this, we are breaking down a very recent but highly cited paper in longevity science called The Biomarkers of Aging for Identification and Evaluation of Longevity Interventions. This paper was published in Cell in 2023, and it's basically the blueprint for what an aging biomarker would look like and how we can use them as real clinical tools.
Yeah, and what's even more exciting is that we have one of the first authors from the paper itself, Dr. Jesse Poganik. Jesse is an instructor at Harvard Medical School and co-director of Biomarkers of Aging Consortium. Welcome to the show, Jesse.
Thank you for having me. Happy to be here.
Jesse, before we dive a little bit deeper into the paper today, why don't you tell us a little bit more about your work and the consortium in general? What is your research?
Well, I would say that my research really focuses around trying to understand the fundamental biology of aging. And for exactly the reason Raghav said at the beginning, if we can't measure it, we can't improve it. We focus a lot on how we can measure aging quantitatively. And so that brings us to the biomarker focus, which I know is something that both of you know a lot about as well. We created this Biomarkers of Aging consortium a few years ago now because at the time, there was really no coordination within the field to try to advance these really exciting tools that a lot of different groups are creating and using. But there's been less effort, coordinated effort anyway, to get these validated and into the clinic, which is where I think a lot of people hope that they will end up someday. And prove to be useful there. So that's how we got the consortium started. And really all of my research focuses on using these biomarkers to try to understand aging and hopefully at some point improve it for people who are aging.
Yeah, no, the consortium itself has been amazing. Having participated in many of the consortium meetings myself, it's so exciting, and Maxwell has been at many of them as well. It's so exciting that we finally have sort of a group of people that are collectively working towards the same goal in a way. But I just want to take a step back and kind of reiterate some of the points that you're saying, that why this is such a huge effort and why do we really need to build these build this consortium. So Jesse, could you actually shed a little more light on that? Like, why do we really need to have this huge effort around sort of the consortium and around the biomarkers? Like, why are these biomarkers so necessary in some ways?
Yeah. Well, I would say that the biomarkers are necessary because, we need to be able to, quantitatively and in an unbiased way measure aging and the rate of aging in people and in experimental systems. I mean, this is the basis of how you do experimental science, right? But it's also the basis of how you might evaluate, for instance, an intervention that you would give someone to try to improve their health span or their longevity. So that's where I think a lot of the kind of future potential of these biomarkers lies. And I know that's one of your key areas of research, Raghav, using these biomarkers to try to assess the effect of longevity interventions in clinical trials, because as we all know, using an endpoint like lifespan is extremely difficult in humans because we simply live too long. These tools are very useful because they, in the research context, they're very useful because we can use them to quantify the biological age or the rate of aging in model systems. We can understand how different experimental manipulations and interventions in model systems affect aging, affect lifespan. And because most of the biomarkers that we focus on with the consortium are based on omics measurements, there's a lot of rich biological data that underlie what we ultimately turn into a number or a series of numbers representing typically biological age in the underlying data. So they're very, multifunctional, useful tools. And beyond the lab, beyond model systems, we're hoping that at some point they are going to be used in clinical contexts. And that kind of ties back to your original point about why do we need the consortium? We need the consortium because being able to do all of the steps going from systematic evaluation to systematic validation to things like regulatory qualification, that really will never be the work of a single researcher or a single research group. That really has to be a community effort, because the breadth of expertise that you need to be able to do something like that spans from basic laboratory scientists to clinicians to people who have expertise in the regulatory world. So it really, we really realized that there was this need to bring these people with different expertise together in this community to be able to try to advance these tools to the clinic. And the nice thing that we found was actually once we started talking to people, even very senior people within the field, there was a pretty universal recognition for the need for this type of thing to exist. And so that's why, we were able to get a lot of people on board, a lot of really the top experts in the world, I would say, and really rapidly start to take steps to reach this eventual goal.
Yeah, I remember a few years ago going to conferences in aging, and as researchers, we couldn't even come into an agreement to what aging was, or what is the best thing. So I guess this sort of like ties up everything together, because it's getting the expertise and looking instead of like a one thing at a time as a combined event. So yeah, that makes a lot of sense. And so let's talk a little bit more about what are the types of biomarkers that there are, because here I think is where people often get a little bit confused, thinking that they're only observed or quantified based on what happens inside the body, or like looking at omics, for example, as you mentioned. But this is just really one piece of the puzzle. So could you maybe explain a little bit more about the different types that you have mentioned in the paper?
Yeah. Yeah, we can certainly do that. So as you say, there are many different types. One of the first things we do in that paper was that we worked with that group of co-authors, and as you can see, it's about 30 different real leaders in the field, and we spent quite a lot of time, as you might imagine, making that first table of consensus definitions, which are working definitions. They're not to say that they will never change, but the first thing we recognized that we had to do was to be able to have a common language to be able to talk to each other. And one of the definitions is for a biomarker of aging, I should say. And strictly speaking, a biomarker of aging is anything that changes predictably over the course of aging that you can measure. So it means that there are many different categories, as you say. So some of the ones that people might be familiar with are some of the clinical biomarkers. These are things that are measured routinely during blood tests that you might get just at your annual physical exam. Then you have the omics biomarkers, and this is a field that the three of us and many others are involved with day to day. These are biomarkers that are based on these kind of advanced omics measurements where you're taking hundreds or thousands or millions of observations of different molecular features, could be proteins, could be CPG sites in the epigenome, and using a composite model to model aging and predict biological age, rate of aging, etc. And another subset that people may kind of increasingly be aware of are these digital biomarkers, and these encompass the things like the wearables, like your Apple Watch or your Oura Ring. And so there have been some examples. It's probably not quite as developed as the omics subset, but there have been some examples, and I think we will see more of these in the future, where biomarkers are being built based on these wearables. And the nice thing about the wearables, of course, in contrast to the omics, is that these are constant observations, so they can give you kind of real-time feedback if you can make a model of aging based on those data. So that's just a subset, but as you say, there are many different kinds of biomarkers of aging.
Yeah. No, I personally, as you said, we've been working in the omics field, but the idea of sort of wearable biomarkers is really exciting because of the continuous monitoring, right? And the ability to sort of monitor on a daily, hourly basis, and then even be able to sort of very equivocally track interventions in that process, right? And how they might expecting. That is really powerful, in my opinion. And actually, that's just really interesting to me about around the classification of biomarkers of aging, right? So this is kind of one way to classify, where we're essentially classifying the biomarkers from the source we're getting the information, right? Whether it is sort of molecular information, or whether it's more functional or clinical information. But Jesse, I know in the paper you talked about essentially other types of sort of categorizations that might exist based on the clinical applications of these biomarkers, right? Could you talk a little more on that front?
Sure, yeah. And I think this is an important point. It's a little nuanced, but it's something that I would say that a lot of us, myself included in the field, are guilty of conflating sometimes with these biomarkers. So, it's often the case in the biomarker development world anyway, that, you build a nice biomarker and you make assumptions about it. But yeah, what we tried to do in the paper was to make the point that these biomarkers can be used for different purposes. And it's entirely possible that, some biomarkers have properties among these different things. But just to go through them quickly, so you can have predictive or prognostic biomarkers. So this is the type of biomarker where you would try to predict a future event. So that could be mortality, that could be an aging-associated disease. And many of the omics biomarkers, I should say, that we have, the best ones that we have, are considered the best ones because they are the most predictive for, important phenotypes of aging. Typically, mortality is the one that's used. So then you also have a subset of biomarkers that are called response biomarkers. And these are the biomarkers that would be most useful in understanding the response to geroprotective or aging targeting interventions. And Raghav, you've done a lot of work, I know, trying to understand the response properties of the existing biomarkers that are out there. But the reality is, we need to continue building the understanding. Your efforts are, I think, an important step in that direction, but we do need to really systematically understand among the biomarkers that we have, which ones are most appropriate to monitoring responses to geroprotective interventions. And then you can also have discovery biomarkers, which are used to kind of feed back to the process if we're thinking about interventions, where we might be able to identify new interventions using these biomarkers.
And I guess, like, one point, going back to the wearables and using those as biomarkers, it's nice in the sense that, you're constantly tracking someone. So yes, you're getting more data, so you're likely to become more reliable in that data. But at the same time, there's so much data that one
Yeah.
now starts having the problem that how you filter through the noise
Yeah.
and how you filter through all these, peaks that may or may not be true,
Mhm.
right? So it's not as simple as one may imagine, but this just brings back to the point that not all biomarkers are created equal, and just because something changes with age, it doesn't mean that it's going to be a useful biomarker. So what actually makes a good biomarker? What are the points that make it like this is it?
Yeah. Yeah, it's a good question. And, I think the kind of cop out answer is it depends on the application. So, the best biomarker for measuring the response to an intervention is a response biomarker. The best biomarker for predicting future outcomes is a predictive biomarker. And these may be the same biomarker, they may be different biomarkers. So, that's why building an understanding among among these different properties of the biomarkers is really important. But there are some general principles, and this kind of predates our work with the aging biomarkers. For instance, so we tried to extend a lot of these frameworks to be specific to the aging biomarkers. One of them related to the last thing we were talking about, the categorization, is this framework called the FDA BEST framework. But there are also frameworks that kind of describe what makes a useful biomarker. And these are things, they're kind of practical considerations, some of them. So for instance, biomarkers should be accessible. They should be easily measured. So, getting a biomarker that requires a brain biopsy is not a good biomarker because it's not very accessible. So they should be relatively non-invasive, able to be repeatedly measured, reliable, things like that. So there are, all these things that we need to be thinking about. And we kind of go through that in this paper, but actually even more so in the validation paper, which is what followed this paper.
Yeah. And Here's the interesting thing. I don't know if you know this, Jesse, but actually, in like sort of in the I think season finale of sort of Keeping Up with the Kardashians, they try to measure some of their biological ages using these biomarkers. But here's the spoiler alert: none of the biomarkers that they actually used or have been sort of used in these contexts, not just the Kardashian approved ones, but others, sort of satisfy all of these different criteria that you guys came up with, whether it's around responsiveness, robustness, feasibility. A lot of our viewers are not just scientists, but they are sort of people who sort of look up to scientists for sort of advice on what to use in clinic for their own health. Uh, and a question that is often asked is, can we actually use any of these biomarkers right now in clinical setting, right? There are a multitude of companies sort of trying to do this, where they are sort of selling biological age tests using different types of omic, functional data, so on and so forth. And some even go even as far as recommending supplements and other interventions, not naming names here. But what are your thoughts about this kind of ecosystem that has existed now or has come into existence, where we're looking at biological, like the access of biological aging tests is there, as well as potentially the ability to intervene using these biological age tests? What are your thoughts about sort of generally this ecosystem?
Yeah. So there's, of course, there's a different benchmark, right, for that needs to be met for direct to consumer versus what I've been talking about as a clinical application, meaning something that you would get in your doctor's office, right, or as part of a clinical trial. So we're certainly not at the level right now of, where these things would be used in a doctor's office. Now, that's not to say that you don't have very conscientious, well attuned to the science clinicians out there now. But of course, it's a mix. So, what I do say when people ask about, should they test? I mean, short answer, probably no. But longer answer, it's not my position to tell anybody what they should do.
Mm,
But I do think it's important to understand the limitations, right, at this moment in time right now. And I think the big limitations are, number one, we don't really understand how individually useful these tools are. So
yeah.
we don't have a good understanding of the sort of factors that introduce technical variability, or even the factors that may truly influence these biomarkers, may truly influence biological age, but may just be part of the normal experience of life. I mean, one example that we've published before, and has been now validated by multiple groups, is the example of pregnancy. So
Yep.
we've shown that the several of those biomarkers are accelerated over the course of pregnancy and then tend to return back to baseline afterwards. So we don't have a good understanding or a catalog of these kinds of changes that may be affecting the biomarkers in a way that may or may not be clinically actionable. And that kind of leads into the other big issue that I would point out, which is that, and this is something that I should say comes directly from our clinical colleagues working with the consortium. They make this point all the time, which is that it's great that we can measure the biomarkers. It's great that we can estimate biological age, but they don't have anything that they can do with that.
Mhm.
And so
Yeah.
that goes, kind of to this issue that we don't have an established intervention, that in humans that extends lifespan unambiguously. In contrast to mice, where we can very easily do that experiment, and we have done with many interventions, right? So that's a challenge, and I would caution anyone is considering taking advice on what to do with a biomarker result at this point in time. Now, that being said, I do think that the landscape is changing a bit. You might be aware there was a study that came out a few weeks ago from Switzerland, and this was a large clinical trial looking at omega-3 supplementation plus or minus some other things like
Yep.
vitamin D and exercise.
Yep.
And that this was a very nice study because it was very large. Raghav, you know better than anybody that a lot of these intervention studies are at the pilot stage, so they're
For sure.
small groups of people, which is of course where you have to start. It's not to criticize; it's just to say that at this moment in time, this is the situation, and
Yep.
this is the evidence that we have. But I do think that we're going to start to see more of these larger scale clinical trials where larger groups of people are tested with different interventions. And so at some point, what I hope is that the lines will sort of connect where, on the one hand, the validation of the biomarkers proceeds. On the other hand, our understanding of how we efficiently target aging proceeds. And hopefully we get to a nice kind of synergistic point where the understandings converge, and then we really be able to help people. And, exciting efforts like the X Prize, health span, and things like the Prosper program with ARPA-H, I think those are going to be things to watch that really have the potential to be very transformative in achieving that goal.
Yeah.
Yeah. And I think like also just extending on that point, the smaller clinical trials, as you said, is not to be disregarded because, at the end, every data adds up. And, when you put them all together, if all of them are going in the same direction, you end up with a big enough number, which you could somewhat consider as being a large cohort, right?
Of course.
So, yeah, absolutely. But I think like what is really the point here is that this paper in general and all those points that we have touched on is really a wake-up call that if we want to build a future where we can track biological aging, like we track, let's say, our cholesterol levels, we do need to invest in more validation and more trials and more testing. So then, at some stage we will reach this concrete idea of what works and what doesn't. So yeah, that's the future looks good. Yeah.
Yeah,
And,
I absolutely agree.
Yeah, and I think it's really exciting in some ways because the possibilities of it are huge, right? Whether it's the personalized longevity plans that could be implemented, assuming all of these biomarkers work out, the rapid testing of sort of anti-aging therapeutics, sort of the real-time feedback, kind of what we were talking about with wearable biomarkers, and whether sort of your sort of lifestyle changes are working or not. We're not there yet, of course, but this paper hopefully lays that groundwork that we need to get to that point. And the full field is moving fast. And if biomarkers deliver the aging biomarkers that we have been working on for such a long time, they could actually unlock the next generation of sort of a preventative medicine paradigm, which is ideally where we'd like to be at.
Yeah, that's certainly the hope.
Yeah, well, we're just thankful for researchers like you, Jesse, for doing the groundwork and, building those consortiums and, bringing the aging community and research community into a consensus. So hopefully we'll lay the foundations for a preventative gerocentric paradigm of medicine. So thank you so much. And thank you for coming in.
Absolutely, yeah, and thank you guys not only for also doing research, which is very important, but for bringing this to people, because I think not enough people know that this kind of science is going on. So I appreciate that you're doing this effort to bring awareness to more people.
Yeah. No, if you like this episode, hit that follow button and leave us a review and share it with your favorite longevity enthusiasts. And of course, check the notes that we have in the end of each of these episodes for sort of the bonus reads on these different publications that were mentioned in this particular podcast. And thank you for joining us at Decoding Longevity Podcast. See you next time with some really exciting science.